Hello, my name is Piv and welcome to today's webinar on the introduction to EMBL EBI resources. I'm joined today by my colleague Andrew Cowley, who will be assisting me today. So moving into it, the aim of this session is to introduce you to the work of EMBL European Bioinformatics Institute. I'm going to lead you through an exploration of the data resources and tools available, and at the end we'll show you where you can get additional training and support. So I really wanted to start with explaining what and who EMBL EBI are. So the European Molecular Biology Laboratory, which is based at its headquarters in Heidelberg, is a centre of excellence and big molecular biology research centre. We have a number of sites across Europe which specialise in various aspects of molecular biology. There's two based in Hamburg and Grenoble in particular that are based in structural biology. And here at the European Bioinformatics Institute in Hingston, Cambridgeshire, in the UK, we specialise in bioinformatics, services, tools and resources, and some research too. Now, EMBL is formed of 24 member states. We also have a couple of associate member states outside of Europe, that's Argentina and Australia, and we also have the prospect member states, Poland and Lithuania. We have people across 80 different nationalities working for EMBL, and we also have over 1,800 personnel. Now, what does EMBL European Bioinformatics Institute actually do? So one of the things we do offer is scientific services. As well as this, we also have a number of research teams who also develop a couple of services, but specialize in various aspects of molecular biology research. Now, one of our mission statements is to also provide training for scientists across the world, as well as on-site. We also engage with the industry program and have a number of partners across the world. Now, lastly, we also have the Elixir node and hub based within the European Bioinformatics Institute. And this coordinates bioinformatics training capacity and kind of the infrastructure within Europe and across the world. Now, the EBI offers many different resources across the kind of biological span. So we have resources that specialize not only in genomes and nucleotide sequences, but we also go into the metagenomics resources. We also provide functional genomics such as Expression Atlas and Array Express. We have protein sequences, families and motifs, molecular structures that span proteins as well as chemical biological structures. And we integrate all this into systems such as Intact, Reactome, biomodels. Now, there's a number of databases that feed directly into these and they all interact with each other. And I'll be going over this in a bit more detail later. Importantly, all of these databases can integrate in various manners and are linked via the literature and ontologies services, such as Europe PMC and BioStudies. I did mention that we also have a number of research themes. Now, these can go into genes and gene expression. So you can see on the top left there that we have Nick Goldman's research on phylogenetic trees and DNA storage and how to represent these phylogenetic trees. Janet Thornton's research is largely based around structural biology and at the moment the structural biology of aging. We also have systems research headed by people such as Evangelina, and this is studying large-scale signaling networks and tying to all the data that's being produced by scientists all across the world. Now, I mentioned training as one of our kind of important kind of mission statements, and we have three kind of types of training we offer. First of all is on-site. So we deliver courses on-site here at the EMBL EBI in Hingston. And these can range from kind of small courses and workshops to kind of week-long sessions that bring scientists from across the world to us. We also host off-site training. So this is where we will take trainers from across our various resources, services and tools and send them across the world to deliver training on particular resources and aspects of resources. Last but not least, we also have a huge online training database where we kind of offer the ability for people to sign up for free, as well as use train online resources that will take you through quick tours, as well as more in-depth kind of dives into research and, well, the various services that are offered. And I'll be showing you a last slide on this towards the end. So, what do we do? More importantly, the first step is actually generating data. So this is from kind of any users across the world doing any kind of biological research. They'll probably come to a point where they'll be submitting their research as part of the data sharing and standards across the journal they're submitting to. Next, it will get archived, and we have kind of a number of curators and archivists working within the EBI. Now, after this initial kind of archiving classification, it's then available to share within the themselves. Now, a lot of the databases also pipeline into some analyses so that they can be shown and visualized on various other services in a way that is easily studyable within a given tool. So there's also a development phase. So we also have the ability to use these resources via our API, and many of our services do offer this. And the most important part is that you can reuse all the data and use it for your own research. And this is cyclic, so you can use the data that has already been generated to generate your own analyses and data to be put back into a database. Now, I did mention data submission. It's one of the most important aspects of what we do here. We do promote open science, and this is very important as submitting your data allows other people to use your data for whatever they're doing. Now, sometimes a given question can be kind of answered in a number of different ways, and your method may provide one insight, and another person might have another idea and use your data as part of that to study something further. As I mentioned, publication in many areas requires now the provision of data and the submission of data during the submission process for the articles themselves. Now, I did mention the data resources earlier. I also mentioned that the data in the various databases interact in a number of different ways. So, for example, if you start off with the European nucleotide database, the archive, it can often link out to uniprot protein sequences, and from there you can access a variety of different types of information. Sometimes you are able to kind of go into visualizing three-dimensional protein structures within the protein data bank. Now, this wheel on the right-hand side just shows you a glimpse of some of the databases and how they interact with each other. The arcs and the number of arcs tell you how many different kind of database interactions there are, and the width of the arc gives more information about how much of that data is going across. We also have data on a variety of different species, spanning single-celled organisms, bacteria, simple nematode worms, all the way up to invertebrates, vertebrates, and insects. Now, the way biological research is carried forward these days is changing rapidly. With increases in computational power, we're seeing that there's an explosion of data, as well as a variety of different types of data now being integrated into various formats and outputs. And this is really leading into kind of a systems biology approach for understanding huge systems. We're also seeing the application of biology in various different fields and global challenges such as molecular medicine, personalized medicine, agriculture and food security, as well as the environmental sciences. Now, one thing worth mentioning is one of the projects happening across the world called the Human Cell Atlas, of which some of which is happening at the Wellcome Genome Campus, which is looking to basically map every single cell in the human body and its kind of functional outputs. So this is proteins as well as small molecules. Now this is a huge amount of data, and this data will be kind of available to access for free via the various databases curated and annotated within the EBI. Now hopefully this data will translate to not just kind of basic research outputs, but also help clinicians with treating and diagnosing various diseases at different stages. Now there's a lot of demand for data these days. You may have heard the kind of buzz term data and data science. Well, we have scientists at over 3.2 million unique IP that use the services within EMBL EBI. As well as that, we have a lot of kind of data compressed and ready to use. For example, the European Nucleotide Archive has 3.2 petabytes of data compressed, and this just makes up a small amount of the data that we actually look after here at the EBI. So this is just a quick example, a snapshot almost, of the variety of users across the world that use EMBL EBI services. So you can actually visit the live map by going to www.ebi.ac.uk/about/our-impact. And this just shows you just the breadth of kind of users across the world that use our resources. So switching gears now, told you a little bit about how EMBL EBI works. I'm going to go into a bit more detail about how to actually navigate the website itself, starting with the main page. So the main page can be visited by going to www.ebi.ac.uk, and it will take you to this main page here. On the main page, you can see the mission statement and the 5 different aspects that we kind of work on. As well as that, you can see a EBI search tool, and this search engine is very powerful, and I'll be going into a bit more detail about this in a moment. I've highlighted a section here to allow you to easily find any tools or databases that you may or may not be interested in. So if you do click on this button, it takes you through to the tools and databases section. Now, the highlighted windows here on the right hand side allow you to either search through the various tools, which are on the left hand side of the page, or the databases in the middle of the page. Now you can scroll up and down and look for the resources themselves, or go through the A to Z list. Or if you have an idea of the kind of resource you're after, you could try a hand at searching. So if you were interested in, say, proteins, if you type in protein, then you will find those resources most applicable to the databases, such as UniProt, for example. You can also browse them by type. So on the right-hand side, you can see they're divided by DNA and RNA, gene expression, literature, cross-domain, for example. Now, going back to the landing page, we can also see there's a section on depositing data. So if you were to click right here, it takes you to the data submission page. Now, it will look slightly different to you when you first visit this page. And that's because when you first visit yourself, you have the option to answer some questions about the type of data you would like to submit. And from there, a few more questions are asked. And at the end, you'll be given kind of a quick guide to which database you should be submitting to. So for example, here, if we go through steps 1 to 4, it might suggest metagenomics is the best submission option. Now on the right hand side here, you can see the number of different data repositories you can submit to. You can also click on those straight away if you already know which archive you'd like to submit to. Okay, so we're going to jump forward to the EBI search. So on the main page here, you can see the search bar. So this search bar not only allows you to access and search and quickly find information within the EBI website itself, but it also acts as a search engine for biological data. Now, this data is biocentric. So if you, for example, type in something like BRCA1, which is a well-known genome protein, and click search or press enter, it takes you to a search result page. Now, importantly, on the top here, you can see it shows 42 results out of the over 55,000. And you can see on the left-hand side here that you can filter the results in a number of different ways. So if you are interested in genomes and metagenomes, you can click on that section and it will only show results relating to that. Similarly, if you're interested in molecular interactions, you can click on that and go further into the various resources such as Intact and Mint. Importantly, you can see that there's a genome protein summary at the top here. Now, this isn't shown for every protein and gene, but most will have one, and you can see it's separated by organism. So let's click on that and see where it leads. From here, we can see the genome protein summary for human BRCA1. So you can see on the left-hand side here that there are various different tabs that you can click on. So the gene, the expression profiles, proteins, the protein structure itself, as well as the literature section. So in the first section, which is gene, you can already see that there's kind of information relating to exactly where BRCA1 is positioned within the chromosome, and a number of different variants and orthologs. And if you click on these, you are able to link out to the different databases that take care of this kind of information. Now, you can also see highlighted here that you can view this gene in a lot more detail using Ensemble. If we click on expression now, this will allow you to view the expression profile of BRCA1 across various different tissues. And you can also link out to this separately in Expression Atlas if you wish. If we head to protein, information from the UniProt database becomes available, as well as Interpro, which will give you some information about the architecture of the protein. As well as this, if you're interested in protein structures, PDBE's service will allow you to visualize these protein structures and gain some information about the quality of the structure, and you are able to download these from the PDBE site quite easily. Finally, you can read the latest papers as well as look at certain reviews by clicking on the literature tab within this EBI Search protein summary. So EMBL EBI's training has existed for kind of over 10 years, and in September 2017 it marked just over 10 years of EBI training being available. So to mark this, a treasure hunt was created, and you can go through to the Train Online website and access this treasure hunt. Now, this treasure hunt is designed for you to basically go through the various different resources and try and find clues to find, well, all the different clues. It's quite fun, and it's a really good way of seeing how the different interactions of the databases occur. And if you're interested in this, you should definitely check it out. A link is provided at the bottom there. Now, towards the end of the section on EBI Search, I did mention literature services. And one important aspect of that is Europe PubMed Central, also known as Europe PMC. Now, this is kind of similar in kind of terms of PubMed, but offers a lot more in way of the annotation and the linking out to various resources. So you can access it via europepmc.org. And doing so will allow you to kind of get a lot of different benefits. So, for example, you can search for more than just the abstracts. You are able to look at various grants, full texts, guidelines from kind of various kind of government bodies, as well as linking to the public databases that include Wikipedia, as well as the ones I mentioned earlier, such as UniProt. You can also link publications with ORCID profiles. Now, this is a unique identifier for authors, so even if you change name, your profile number stays the same. And you can easily search the kind of content of a particular researcher by clicking on their ORCID number. As well as that, there's also an available service such as SciLight and external links services, and you can link these to RSS feeds quite easily. So let's dive into a quick search. If you were to go to europepmc.org and type in CD44, which is a cell surface receptor, and hit enter, you'll be taken to a results page. Now, the first 25 most relevant results are usually shown, and sometimes you can get quite a few results, but importantly, you are able to filter these through as well. You can see, for example, that you can sort by relevance, the date, the times that a given article has been cited, so the citation number. As well as that, you can choose to look at open access papers only, or looking at reviews, patents, books, and documents. Now, importantly, you can also use the annotation services offered within Europe PMC. So if we take this paper as an example, if we were to locate this paper within Europe PMC, actually click on the page so you see the abstract. If you click on the section on the right-hand side, which has a number of checkboxes, you can see there's annotations available in the abstract. So clicking on, say, diseases, for example, will allow you to find keywords within the abstract that will relate to a certain disease. As well as this, they also offer annotation of chemicals, genes, proteins, and other Go processes. This can be very useful for quickly skimming a paper and finding the relevant kind of areas that you wish to study further. Another great feature of Europe PMC is the ability to click on certain tabs, such as the Bioentities tab. Now, if we take bioentities, we're able to quickly go to the various proteins and gene names mentioned and export them directly to formats used in other data kind of analysis platforms. Importantly, you're also able to export a number of various links using the export tab that's often on the top right of a search page. So these can be exported as .csv files, for example, or if you're interested in putting it into your own reference management software, then going into something such as a bibtex file also works. So that's a brief tour of EMBL EBI, as well as the search tool, and Europe PMC and some of its resources. In terms of additional training and support, the EMBL EBI's Train Online feature is fantastic for taking short courses and learning about the various aspects of biology. So for example, we have a number of tutorials available that will go through various resources in a lot more detail. We have webinars such as this one that are stored on Train Online. Quick tours are essentially a 30-minute or less quick tour of of a particular resource, and it's given as a quick guide for you to access content quite easily afterwards. Now, I did mention conceptual courses, which will just go over the basics of certain aspects of biology. You can access this without signing up, but if you do sign up, you can access quizzes, track your learning. As well as this, you can pause halfway through a particular course and then pick it up afterwards, and be the first to know about any upcoming new courses and webinars in the process. So most of our resources, services, and tools also have a help and contact button or page available for you to click on and speak to the help desk. Now, the thing we offer is most of the help desk is kind of seen by the curators and the developers within a given resource and team. So you will be talking to a member of the team and get help where needed. We're also available on social media. So if you want to follow updates on training courses and events and see what we're up to, you can follow us at EBI Training. If you want updates on general news from the EBI, that's via at EMBL EBI. We also have a list for you to kind of access and have a look at, and that includes people within the EMBL as well as the EBI. And if you want to follow specific updates from the various resources that you know you're interested in, such as Uniprot, Ensemble, and ENA, you can do so as well. There's also a Facebook page available. That concludes this whistle-stop tour of the EBI resources. Thank you very much for listening. And if you do have any questions, please let us know and we'll be happy to answer them.